everyone, it's Mrs. Shannon. Welcome to the first and many screencasts that you will see this year in biology. Rather than take time in class to do lectures, you're going to find that a lot of them are going to be done in this way, in which you are given the notes in a video format. You'll also have a copy of the notes and slides that you can go back to. But this will allow you to stop, pause, take time to write things down, maybe do the lecture in different chunks of time depending on how much time you have at home. And you can watch it on a phone, electronic device, at school, whatever the case may be. When we do notes, I am going to show you at the beginning here Cornell notes. Some of you are used to Cornell notes. You did it in middle school. Some of you aren't. But it's a good habit to be in. Cornell notes let you take notes in one part of your note paper and then use the other part to go back and add to it. And so oftentimes they're called two-column notes too, where you have the title of the notes and the date, and then there's a line drawn down. And on the right side, you take any notes during presentations, you underline keywords. Skip a line if you're writing it out, just so you have space. Make sure you abbreviate. Don't try to write word for word. When you are done, you will then later go back and answer study questions. And there will be different levels of study questions to answer. Make sure that you can do that after you're done with the notes. And then at the bottom, you're going to reread through your notes and write a summary. The summary should be 3 to 5 sentences. Now, to get you started, I will tell you that a lot of the screencasts will have notes already printed up in Cornell style. So here's an example of notes for what we're doing today, scientific method. As the lecture is going on, you are going to read and fill in the blanks. When you are done, you are going to go back and answer the questions. And these numbers refer to how sort of tricky they are. With DOK 1 with level 1, depth of knowledge, being kind of what the easiest question is, and then they get a little harder as you go up or go down here, 1 through 4. When you're done, you'll write a summary. Now, this is a long lecture. I don't usually like to do them this long, but I think for the first one, it sort of all goes together, and it will. So if you need to take a break from it, I understand. Take a break, know where you left off, and then come back to it. So what we're going to do here is I am going to put this in full screen. In just a moment, you present, and we'll go through it. Okay. When you are done with your notes, these notes are going to be going into your interactive notebook. I expect that you will study the notes. That's why you're taking them. Quiz yourself with them. Review your summaries. When we come up to unit exams, you're going to want to review them. Understand what you're writing. So write them in a language, your summaries, in a language that you also understand them in. So this is a review on the scientific method, and I'm saying a review because I hope that you in middle school or elementary school talked about the scientific method. It's a method that helps you think like a scientist, record like a scientist, and think back about what you did just like a scientist. And there's different ways that scientific method is taught. There's different numbers of steps, but they all follow the same basic idea. Science actually means to know. It's a Latin word. So you can sound fancy. You can say that you know Latin now because science is derived from the word to know. And it's just a way of asking questions. Pseudoscience, if you've ever heard that term, and especially, in the news, you might hear pseudoscience. It's false science. Pseudo means false. And pseudosciences are supernatural ideas, or they're ideas about religion. We don't study that in science. It's considered false science. We study things that we can ask questions about, and actually find answers and experiment with and collect data. So you'll hear oftentimes laws, theories, and models. A law is a rule that describes patterns observed in nature. For example, the law of gravity that states that when you drop something, it falls to the ground. It describes patterns that were observed. The theory is why these laws might happen. Okay? Or how this happens. And then models I'll use models a lot, are any kind of representation of an object itself, like a cell, or the process of gravity, something falling. And it can be pictures, 3D representations. It can also be graphs. You can write stories, anything to represent something that is not right there in front of you, or that may be too big or too little or too complicated to give in its entirety. Theories and laws are well accepted by scientists, but they are not set in stone. Even though it's a law, it's not set in stone. If there's evidence that comes up that changes it, that's okay. However, to become a working theory, it has to have a lot of support, a lot of evidence to support it. To become a theory, an explanation of how or why, or to become a law even, there's a process that is often undergone the scientific method, and it usually starts with a problem or a question. It's based upon observations. So you see something, you hear something, you smell something, and you wonder about it, and you generate a question or a problem about why it happens or how it happens. And this problem or question is one that can be tested. So you see plants growing out in the sun, and you wonder, what is the effect of sunlight on plant growth? That's a question that can be tested. After you've made your observation and come up with a problem or a question, you have to go into your research. Now, observation is here as well. You're going to be actually observing throughout the entire process. But you're also going to add in research. Not only are you going to use your own your own observations about what you see, about the plant growing, about what kind of sunlight it's in, you're also going to use the internet or books or journals to help give you some research about help what conditions are plants normally found in, how much sunlight do they usually get a day? Because what that's going to help do is help you formulate a hypothesis and an experiment. A hypothesis is a prediction, but it's it's not. A hypothesis is often called an educated guess. It's more like an educated prediction. It's a solution to the problem, but here's the key. It's based on the research. It's based on research, science-based research. It's not just randomly thrown out there. And the way that we're going to be doing it is if, then, because. And I'll say it over and over. If I change something, then I expect this to change. And the because part is the science behind it. So in this case, if sunlight has an effect on plant growth, then plants will grow larger in the sun. Okay, this is the prediction. And then the science, because, whoopsie, if I can go back here, because it is needed for photosynthesis. And photosynthesis is the process of taking sunlight and converting it into glucose. So now that we have a hypothesis, it's an educated guess based on science that is testable as well. It's time to test it. And we test it with a step-by-step procedure. And that procedure needs to be clearly defined, because if we come out with an awesome experiment and we want to share it with others, we need to make sure that it's shared in a way that can be reproduced by others. It is clear and detailed so another scientist can replicate it. And we kind of did that with the Lego activity this school year. We're going to list all the materials that we need so other people can use it. And then we have to identify some parts of our experiment. We have things called independent variables. These are just fancy terms. You might hear about variable in math class, and we have dependent variables. The independent variable is what you or the person experimenting or the investigator changes. Whatever they have control over, whatever they can change. You can change the amount of time that you're testing something, or you can change the temperature that you're testing something out. That's the independent variable. It's also known as the group that's being experimented on. Okay, it's the experimental group. Example, the amount of light being changed. If you change the amount of light different plants are given, that's the amount of light is the independent variable. And the experimental groups are the ones with like 4 hours of light or 5 hours of light or 6 hours of light. The dependent variable is what you're going to measure. It is due to whatever effect this might have. It's due to that. I always say start with a dependent. What are you trying to measure? And if you have to identify it and then work your way backwards. It's sometimes easier when I'm not sure to look for that word measure or look for something that is measured, identify it as the dependent variable. And then the other thing that's being switched will be your independent. So if we're looking at changing the amount of life, it's going to affect the plant growth. We're going to be measuring it in centimeters. Okay, a couple other vocabulary terms that you're going to need to be to know and be able to identify. Number one, we have what's called the control group. The control group, you always want to run a control group because that's what you're going to compare your experimental group to. It's the group or sample that is used for comparison, and that is usually the group that is run with none of the independent variable or normal conditions of it. So in this case, if we're changing the independent variable as amount of light, having no light would be a control group. The constant is everything else that stays the same. So I always think about the environment that experiment is being run in, and whatever's not changing is a constant. So if you're growing them in the same room or the same place outside, if the temperature is the same, if the humidity is the same, if you're using the same type of water, if you start with the same type of plant, those are all constants. And you can usually come up, my minimum, I always say, is come up with 4 minimum constants when we're designing an experiment. Although you should be able to think of a lot more. When you are running the experiment, you're going to be making more observations because you're going to be observing the process as you collect data. The information and the observations can be, during the experiment, can be called either qualitative or quantitative data. Qualitative data describes what it looks like, the color of it, the plants, if it's withered up, if it's gone from green to a brown color. Those are qualitative observations, qualitative data. Quantitative data has that letter N in it. Okay, notice that's the only difference between the way these are spelled. The N, think numbers, numerical measurements. It's data that involves numbers, 15 centimeters, 5 grams, 20 inches. If it involves a number, it's quantitative data. And both are good, although we like quantitative whenever possible. So if we can get a quantitative measurement, if it's possible, definitely go for it. Qualitatives are nice to have in addition. Once we've collected our data, then it's time to try to figure out what the heck it means. And that's when we analyze our results. We usually use a model, such as a graph, to put them in a way that makes it more easy to understand, like visually easier to understand. We'll have a cheat sheet on graphing, but we only use, well, not always, but most often when we set up a graph, we only use one quadrant. So if you're used to the crosses in math class that has all 4 quadrants, we only use number 1. We can do line graph, pie graph, or bar graph. Pie charts, yes, are ways to display the results, but we don't really use them much. If there's numbers, chances are, if the measurements that you're taking are numbers, quantitative data, chances are we'll be doing line graphs. Not always, but that's kind of the rule to follow. Line graphs are going to be the most common ones. If the data you're collecting does not have numbers, then we'll probably do a bar graph. When in doubt, ask. I have no problem helping with that. Precision and accuracy, I'll come back to. Precision is just basically how close the measurements are to each other. You could shoot a bow and arrow, and all 5 times it's at the very outer edge of the bullseye in the same exact spot. They could be precise because all 5 of them are together, but they're not very accurate. Accuracy is how close it is to the actual value. So if they had landed in the bullseye, then it would be accurate. This one right here is very important. Number of trials. When you measure, okay, so with precision and accuracy, you want to be careful on how you're measuring. Make sure that you're precise okay, that they're precise and accurate. But the accuracy is what you want to focus on. If they're not as precise, we have to wonder what's going on. That's a different story for a different day. Sometimes it might bring up other questions, but we really want to focus on accurate data collection. So measure carefully, which is precisely, but collect data in multiple trials. That's repeated tests to increase the accuracy. So the number of trials are going to get us, or multiple trials will get us this true value. Being careful when you actually do the measurement and you look at the numbers, that will give us the precise value that we need. After you've looked at the data in graph form, it's time to start doing a conclusion. The conclusion is the solution to the problem. Remember that problem you had at the beginning, the sunlight affect plants. Well, now it's time to answer that. There are a couple things you need in your conclusion. Number one, you need to state whether your hypothesis is accepted, whether the data supports it. So if it's accepted, the data supports it. It answers it. It says, yes, your guess was correct. Or rejected. You must say my hypothesis or the hypothesis is accepted, or if it's not, it's rejected. That means your data does not support it. And then you have to say why the heck it's important. You summarize and explain the significance of your results. And finally, the third thing that you need to include in your conclusion is sources of error. Finally, when you're done, you're ready to communicate. Remember, communication is one of those science process skills you're going to be graded on. Not only designing the experiment, but communicating your results. So you need to present your information. Sometimes it will just be done in the classroom in a lab worksheet, or sometimes it will be done in slides. You'll do a presentation. But you're going to end up presenting your information to others in some format. And you need to make sure that when you present it, that it's able to be retested by others. Now, in the true real world setting, other scientists would go ahead and verify your results. And here, what we'll often do is have multiple groups do the same experiments to see if their results come up to be the same as yours. So take a break. Ask yourself, do you think you can name all 8 steps? You can hit pause if you want. Okay, here they are. Problem, observation, hypothesis, experiment, collect data, analyze results, conclusion, and report findings. I do want to change, and I don't know if I can do it during this, but I'll probably go back and make the change on your slides. Where it says number 1, problem, it could be an initial question. So it's problem or question. And then down here where it says report findings, that's a form of communication. So oftentimes I'll use that word communicate. Okay, final notes. Depending on where you are looking, the scientific method could be 5, 6, 7, 8, or 9 steps. It depends on who's describing them. The procedure doesn't change, only how steps are divided. Sometimes they combine the results, the analysis, and the conclusion. Sometimes they add in an extra steps where you're going to have to repeat it. Also, the scientific method is more of a cycle. Soon as you think you're done, you realize you're back at the beginning. More to come on this later, but it's like a nonstop process. So I hope you're able to answer the questions and fill in the blanks. If you still have blanks, go back through and find them in the presentation. When you are done, answer the questions on the left of your paper. And finally, you're going to write a few sentences at the bottom. These will be taped into your notebook and you can refer back to them at any time. If you have questions, feel free to ask. Okay, I hope this helps and I'll talk to you in the next one.